Hi class, this is your instructor, Scholar Huff, and we're now in Chapter 29, Seedless Plants. The colonization of land by plants fundamentally altered the history of life on Earth. A terrestrial environment offers abundant CO2 and solar radiation for photosynthesis. For 500 million years, the lack of water and higher UV radiation on land limited green algal ancestors of land plants to aquatic environments. Evolutionary innovations for reproduction, structural support, and prevention of water loss enabled the transition to land. The shift to life cycles dominated by a diploid generation helped protect against damaging mutations from UV exposure that would be lethal to a haploid generation. Long before seeds and flowers evolved, the earth was covered by seedless plants like the moss that covers this tree, and the ferns that you see growing alongside the moss with the tree. Here, we will consider the evolutionary innovations in seedless plants during the first 100 million years of terrestrial life. If I were in your kicks, those are called shoes in some places, I'll be sure to review the visual outline of this chapter on page 611. It shows all the places we're going in a very concise and easy to understand way. 500 million years ago, most of the continents were in the southern hemisphere, and a diverse array of animals swam through the seas, feeding on the abundance of algae. In stark contrast to the terrestrial environment was a barren landscape. Slowly, sea life began to explore the possibilities of living on land. Eventually, land plants evolved from green algae. This began the conversion of bare ground to the rich and diverse terrestrial ecosystems of today. Green algae and the land plants shared a common ancestor a little over 1 billion years ago and are collectively referred to as the green plants. DNA sequence data are consistent with the claim that a single individual gave rise to all green plants. The green plants are photoautotrophic, but not all photoautotrophs are plants, as some microorganisms are capable of photoautrophy. Although the plant kingdom includes green algae, it does not include the fungi, which are more closely related to metazoan animals. Fungi, however, were essential to the colonization of land by plants, enhancing plants' nutrient uptake from the soil. The adaptation to terrestrial living was one of the most significant evolutionary events in the billion-year-old history of the green plants. Here, we will focus on the evolution and adaptation of land plants. Land plants had to develop mechanisms to avoid water loss, to protect from the harmful effects of solar radiation, and to effectively disseminate gametes for reproduction. In addition, although all land plants alternate between a haploid gametophyte and diploid sporophyte generation, there is a trend toward dominance of the sporophyte in the more advanced group of plants. Some saltwater algae evolved to thrive in freshwater environments. Just a single species of freshwater green algae gave rise to the entire terrestrial plant lineage, from mosses through the flowering plants known as angiosperms. The exact nature of this ancestral algal is still a mystery, but close relatives, the charophytes, exist in freshwater lakes today. The green algae is split into two major groups: the chlorophytes, which never made it to land, and the charophytes, which are a sister clade to all land plants. Together, the charophytes and land plants are referred to as the streptophytes. Land plants have multicellular haploid and diploid stages, unlike the charophytes. The presence of a diploid embryo in the life cycle is also an innovation to land plants. As land plants have evolved, the trend has been toward more embryo protection and a reduced haploid stage in the life cycle. Here we are. red algae, chlorophytes, still in the water, charophytes, and now the land plants. So here you all will learn about bryophytes, tracheophytes, and the euphylophytes. And then in the next chapter, you all will then learn about the seed plants. Let us begin. Unlike the freshwater ancestors, most land plants are not immersed in water. As an adaptation to living on land, most plants are protected from drying out by a waxy surface material called the cuticle that is secreted onto parts that are exposed to air. The cuticle is relatively impermeable, preventing water loss. This solution, however, limits the gas exchange essential for respiration and photosynthesis. Tiny mouth-shaped openings called stomata in the cuticle of land plants allow the absorption of carbon dioxide and simultaneously the release of oxygen and water vapor. Moving water within plants is a challenge that increases with plant size. Bryophytes, the most primitive of the land plants, cannot grow beyond a certain height because they lack vasculature to efficiently transport water. Tracheophytes are more advanced, producing specialized vascular tissue for transport over long distances. Xylem is the tissue that carries water. Dissolved nutrients, including sugars produced by photosynthesis, are carried in phloem. Terrestrial plants are exposed to higher intensities of UV irradiation than aquatic algae, increasing the chance of mutation. These land plants minimize the effects of mutation by carrying 2 copies of every gene. That is, their bodies are diploid, meaning that deleterious recessive mutations are masked. All land plants have both haploid and diploid generations, but there is an evolutionary shift toward a dominant diploid generation. Humans and other animals are diplontic, or at least have a diplontic life cycle, meaning that the diploid stage is multicellular. In animals, meiosis produces single-celled haploid gametes, egg and sperm cells, which fuse at fertilization to produce a diploid embryo, which divides by mitosis to become the next generation. The products of meiosis never divide by mitosis, so there is no multicellular haploid generation in animals. By contrast, the land plant life cycle is haplodiplontic. It has both multicellular haploid and diploid stages. Land plants undergo mitosis after both fertilization and meiosis. The result is a multicellular diploid individual, the sporophyte, and a multicellular haploid individual, the gametophyte. The haplodiplontic cycle is a fundamental feature of plants. And you can see this, and I'll call it the alternation of generations, in figure 29.2. Many brown and red and green algae are also haplodiplontic. Humans produce gametes via meiosis, but land plants actually produce gametes by mitosis in a multicellular haploid individual. Meiosis produces a haploid spore that divides by mitosis to produce a haploid gametophyte. Let me go to the figure. This is where I am, or at least this should help you with what I'm getting at. And I'll say again, meiosis produces a haploid spore that divides by mitosis to produce a haploid gametophyte. I hope that you all can follow where I am. I went from that spore mother cell to the haploid spores, to the gametophyte. The multicellular diploid generation, or sporophyte, alternates with the multicellular haploid generation, or gametophyte. Sporophyte means spore plant, and gametophyte means gamete plant. These terms indicate the types of reproductive cells that respective generations produce. The diploid sporophyte produces haploid spores by meiosis. Meiosis takes place in structures called sporangia, where diploid mother cells or sporocytes undergo meiosis, each producing 4 haploid spores. Spores are the first cells of the gametophyte generation. Spores germinate and then divide by mitotic cell division to produce a multicellular haploid gametophyte. The haploid gametophyte produces gametes by mitotic cell division. Consequently, the gametes, egg cells and sperm cells, are also haploid. When 2 gametes fuse, the zygote they form is diploid and the first cell of the next sporophyte generation. This zygote grows into a diploid sporophyte by mitosis and produces sporangia in which mitotic cell division occurs. All land plants are haplodiplontic. However, the haploid generation consumes a much larger portion of the life cycle in mosses and ferns than it does in seed plants. The gymnosperms and angiosperms. In mosses and ferns, the gametophyte is photosynthetic and free-living. When you see a moss, you are actually looking at the gametophyte generation. The sporophyte is a small structure attached to the gametophyte. Although ferns produce photosynthetic gametophytes, they are barely visible to the naked eye. When you see a fern, you're looking at the sporophyte generation. The gametophyte is even smaller in seed plants, typically being microscopic. So as with ferns, the sporophyte in seed plants is the dominant generation. Although the sporophyte generation can get large, the size of the gametophyte is limited in all plants. When the gametophyte generation of mosses produces gametes at its tips, the egg remains stationary, whereas the sperm swims to the egg in a droplet of water. In more advanced plants, the tracheophytes, the gametophyte is composed of only a few cells, and the sperm no longer need to swim to reach the egg. Having completed an overview of plant life cycles, we will now consider the major groups of seedless land plants. As we proceed, you will see a reduction in the size of the gametophyte and the gametangia, the structures in which gametes are produced, with a corresponding increase in the specialization for life on land. Bryophytes are the closest living descendants of the first land plants. Plants in this group are also called non-tracheophytes because they lack the transport cells called tracheids. Bryophytes include the liverworts from phylum Hepaticophyta, the mosses from phylum Bryophyta, and the hornworts from phylum Anthocerophyta. Water and gas availability were limiting factors for early terrestrial plants. These plants likely had little ability to generate internal water levels and tolerated drying out, as seen as in most extant mosses. Fungi and early land plants cohabitated, and the fungi formed close associations with the plants, enhancing water uptake. The beneficial symbiotic relationships between fungi and plants, called mycorrhizal associations, are also found in many existing bryophytes. Approximately 16,000 species of bryophytes are simple but are found in diverse terrestrial environments and even deserts. The gametophyte is small but specialized for light capture and photosynthesis. Water and nutrients are transported through the gametophyte via simple conducting cells. Egg and sperm cells produced by the gametophyte unite to form the sporophyte generation, which typically grows upright from the surface of the ground-dwelling gametophyte. Because the sporophyte is above ground, it can effectively disperse the spores it makes. These spores are carried by air currents, and when they land, they germinate into new gametophyte plants. Scientists now agree that bryophytes consist of 3 quite distinct clades of unspecialized plants: liverworts, mosses, and hornworts. Like their aquatic ancestors, bryophytes require water, such as rainwater, to reproduce sexually. This is also seen in ferns and some other vascular plants, the tracheophytes. The sperm must swim to the egg. It is not surprising that bryophytes are especially common in moist places, such as the tropics and temperate regions. They are commonly found on damp forest floors and by stream beds. The old-time English word wort means plant. Some common liverworts have flattened gametophytes with lobes resembling those of a liver, hence their name, the liverwort. Although the lobed liverworts are the best known representatives of this phylum, they constitute about 20% of the species. The other 80% are leafy and superficially resemble mosses. The gametophytes are flat instead of upright, with single-celled rhizoids that aid in absorption. Although rhizoids function like roots, they are not organs because they are unicellular. Some liverworts have air chambers containing rows of photosynthetic cells, each chamber having a pore at the top to facilitate gas exchange. Sexual reproduction in liverworts is similar to that in mosses. Lobed liverworts may form gambitangia in umbrella-like structures. In this case, the sporophyte is suspended above the ground by the gametophyte. This is different from most bryophytes in that the sporophyte itself forms a stalk above the ground to aid in spore dispersal. Asexual reproduction occurs with lens-shaped pieces of tissue that release from the gametophyte to grow new gametophytes. Unlike other bryophytes, the gametes of mosses typically consist of small leaf-like photosynthetic structures, not true leaves, containing vascular tissue, arranged around a stem-like axis. And that shows that to you here. And the axis is anchored to its substrate by means of rhizoids. Each rhizoid consists of several cells that absorb water, but not nearly the volume of water that is absorbed by a vascular plant root. Moss leaf-like structures have little in common with the leaves of vascular plants except for the superficial appearance of the green photosynthetic flattened blade and slightly thickened midrib that runs lengthwise down the middle. Only one cell are thick, except for the midrib. They lack vascular strands and stomata, and all the cells are haploid. However, mosses do have stomata on the capsule portion of the sporophyte because of that are the basal land group with stomata. Although mosses lack a vascular system, water may be passively carried up the strand of specialized cells in the center of the moss gametophyte. Multicellular gametangia are formed at the tips of the leafy mossy gametophytes. The female gametangia, the archegonia, may develop either on the same gametophyte as the male gametangia, the antheridia, or on separate plants depending on species. A single egg is produced in the swollen lower part of the archegonia, whereas numerous flagellated sperm are produced in an antheridium. Because gametophytes are haploid, eggs and sperm are produced by mitotic cell division, not meiotic cell division, in the gametangia. When sperm are released from an antheridium, they swim with aid of flagella through a film of dew or rainwater to the archegonia. One sperm unites with an egg, forming a diploid zygote. The zygote divides by mitotic cell division and develops into the sporophyte, a slender stalk with a swollen capsule or sporangium at its tip. The base of the sporophyte is embedded in gametophyte tissue, its nutritional source, which is what you're seeing here. The sporangium is often cylindrical or club-shaped. I guess I'll show that to you again. Spore mother cells within sporangium undergo meiosis, each producing 4 genetically different haploid spores. In many mosses, at maturity, the top of the sporangium pops off and the spores are released. A spore that lands in a suitable damp location may germinate and grow via mitotic cell division. Into a thread-like structure, which branches to form rhizoids and buds that grow upright. Each bud develops into a new leafy green gametophyte plant. In the Arctic and Antarctic, mosses are the most abundant plants. The greatest diversity of moss species, however, is found in the tropics. Many mosses are able to withstand prolonged periods of drought, although mosses are not common in deserts. Some mosses, such as peat mosses, can absorb up to 25 times their weight in water and are valuable commercially in potting soils or as fuel when dry. The origin of hornworts is a puzzle. They are most likely among the earliest land plants, yet the earliest hornwort fossil spores date to the Cretaceous period, when angiosperms were emerging. There are only about 200 species of hornwort. However, the hornworts form an important link in the study of plant evolution because they are a sister group to the tracheophytes. The hornworts are likely to provide insights into the transition from the dominant gametophyte generation in the bryophytes to the dominant sporophyte generation in the tracheophytes. The sporophytes of hornworts are similar to the vascular plant sporophytes. Hornwort sporophytes look like green horns when they arise from that flattened structure called a thallus that is usually 1 to 2 centimeters in diameter. The sporophyte base is embedded in gametophyte tissue, from which it derives its nutrition. It has stomata to regulate gas exchange, is photosynthetic, and provides much of the energy needed for growth and reproduction. Mitotic cell division occurs within the horn, and spores are released along its entire length. Tracheophytes, also known as vascular plants, first appeared about 410 million years ago. The first tracheophytes with a relatively complete record belong to the phylum Rhiniophyta. It is impossible to know for sure what the earliest of these vascular plants looked like, and let us now continue. Three clades of vascular plants exist today: lycophytes, called the club mosses; pterophytes, which are the ferns and their relatives, or the ferns and the allies; and 3 seed plants. We will now explore the first two clades. Advances in molecular systematics have changed the way we view evolutionary history of vascular plants. Whisk ferns and horsetails were long to believe to be distinct phyla that were transitional between bryophytes and vascular plants. Phylogenetic evidence now shows that they are the closest living relative to ferns, and they are grouped as pterophytes. Tracheophytes dominate terrestrial habitats everywhere except for the highest mountains and the tundra. The haplodiploid life cycle persists, but the gametophyte has been reduced in size relative to the sporophyte during the evolution of tracheophytes. A similar reduction in multicellular gametangia has occurred as well. The reduction in size of the gametophyte is a major theme in the evolution of land plants. So one thing I shall mention that I did not mention a moment ago is Cooksonia, the first known vascular land plant, which was homoceros, producing one type of spore. Secondly, it became a successful colonizer of the land by developing efficient water and food conducting systems called vascular tissue. So one type is xylem, conducting water and dissolved minerals upward from the roots, and another type, phloem, conducts sucrose and hormones throughout the plant. Given that, let us now continue on. The bryophytes have no tissues. If you remember moments ago, I mentioned they only have cells. By them not having tissues, clearly they don't have any organs. Not to mention, without tissues, they cannot transport water up. Hence, that bryophytes are so short. So fossils of early seedless vascular plants had transport cells in their stems, but had no roots. Well, now these plants have roots. So I'll say true roots are found only in trichophytes. Other, somewhat similar structures enhance either transport or support in non-trichophytes, but only roots have a dual function, providing both transport and support. Lycophytes diverged from other trichophytes before roots appeared, based on fossil evidence. So it appeared that roots evolved twice. Leaves increase the surface area of the sporophyte, enhancing photosynthetic capacity. Lycophytes have single vascular strands supporting relatively small leaves called lycophils. True leaves called euphils are found only in ferns and the seed plants. So lycophils, lycophytes only, euphils, ferns, and seed plants, having distinct origins from lycophils. Lycophils may have resulted from vascular tissue penetrating small leafy protuberances on stems. Euphils most likely arose from branching stems that became webbed with leaf tissue. I don't know why your book is starting with seeds here. Seeds are highly resistant structures, well suited to protecting the plant embryo from environmental stresses and to some extent from predators. In addition, almost all seeds contain a supply of food for the young plant. And I said I don't know because guess what? Lycophytes and pterophytes do not have seeds. They are dispersed via haploid spores. Fruits in the flowering plants, called angiosperms, add a layer of protection to the seeds and have adaptations that assist in the seed dispersal, expanding the potential range of species. Flowers allow plants to secure the benefits of wide outcrossing in promoting genetic diversity. And then before moving on to the specifics of lycophytes, let us continue on here. So if you look closely, we have an illustration. I guess I'll say a quote-unquote cladogram. And what happens here, or at least what you're shown, is land plants, and it says they're innovations. You can even call these synapomorphies, meaning each of these, where they evolve, it shows you that each plant coming thereafter has that same innovation. So this would be something that I would recommend you look at and even study, because this should help you greatly. The earliest vascular plants lacked seeds. Members of 4 phyla of living vascular plants also lacked seeds, as do at least 3 other phyla known only from fossils. So as we explore the adaptations of the vascular plants, we focus on both the reproductive strategies and the advantages of increasing complex transport systems. The lycophytes, called club mosses, are the sister group to all other vascular plants. Despite the early divergence between lycophytes and all other vascular plants, there are similarities among the groups. For example, leaves developed independently in the lycophytes and other vascular plants. Lycophyte leaves are small, unbranched veins, whereas the higher vascular plants produce larger leaves with complex pattern of branched veins. Some ancient club mosses looked like trees we see in the seed plants of today. So the tree-like form is another example of the interdependent origins of similar features in the lycophytes and other vascular plants. Today, club mosses are a worldwide in distribution and are most abundant in the tropics and moist temperate regions. However, modern club mosses are small. None have tree-like growth form of their ancient ancestors. Club mosses, they resemble true mosses, but their internal vascular structure and reproductive processes became known. It was clear that they are unrelated to mosses. The sporophyte stage is the dominant stage. Specialized leaves bear spores, which are produced via meiosis. In most lycophytes, the haploid spore germinates into a small, free-living gametophyte that produces both sperm and eggs. The gametophyte of some lycophytes is photosynthetic. After fertilization, a small sporophyte remains nutritionally dependent on the gametophyte until its green leaves develop and start to carry out photosynthesis. The phylogenetic relationships among ferns and their near relations is intriguing. A common ancestor gave rise to two clades. One clade diverged to produce a line of ferns and horsetails. The other diverged to yield another line of ferns and whisk ferns. Whisk ferns and horsetails are close relatives of ferns, like lycophytes and bryophytes. They all form antheridia with their sperm and archegonia containing their eggs. Water is required for the process of fertilization, during which the sperm have flagella, swim to, and unite with eggs. In contrast, most seed plants have none flagellated sperm. In whisk ferns, which occur in the tropics and subtropics, the sporophytic generation consists merely of evenly forking green stems without roots. The 2 or 3 species of the genus Psilotum, such as Psilotum nudum, do, however, have tiny green, spirally arranged flaps of leaf-like tissue lacking veins and stomata. Given the simple structure of whisk ferns, it is particularly surprising to discover that they are monophyletic with ferns. The 15 living species of horsetails are members of a single genus, Equisetum. Fossil forms of Equisetum extend back 300 million years to an era when some of the relatives were tree-like. Today, they are widely scattered throughout the world, mostly in damp places. Some that grow among coastal redwoods of California may reach heights of up to 3 meters, but most are less than a meter tall. A horsetail sporophyte consists of ribbed, jointed photosynthetic stems that arise from branching underground rhizomes with roots at their nodes. A whorl of non-photosynthetic scale-like leaves emerges at each node. The hollow stems have silica deposits in the epidermal cells. Horsetails are also called scouring rushes because pioneers of the American West used them to scrub pans. Cone-like structures on horsetail sporophytes produce haploid spores via meiosis. Spores are carried away by air currents, and when they land on the ground, they germinate into photosynthetic gametophytes. So each gametophyte produces both eggs and swimming sperm, which unite at fertilization. The young sporophyte is still dependent upon the gametophyte until it produces photosynthetic tissue. Ferns are the most abundant group of seedless vascular plants, with about 11,000 living species. Research indicates that they may be the closest relatives of seed plants. Rainforests and swamps of fern trees growing in the eastern United States and Europe over 300 million years ago formed the coal currently being mined. Today, ferns flourish in a wide range of habitats throughout the world. However, 75% of the species occur in the tropics. The conspicuous sporophytes may be less than a centimeter in diameter or more than 24 meters tall, with leaves up to 5 meters long in the tree ferns. Shown to you, one is here. The sporophytes and much smaller gametophytes, which rarely reach 6 millimeters in diameter, are both photosynthetic. The fern life cycle differs from that of a moss, shown for you all here, and we will review this in class a bit more. The independence and dominance of the fern sporophyte. The fern sporophyte is structurally more complex than the moss sporophyte, having vascular tissue and well differentiated roots, stems, and leaves. The gametophyte, however, lacks the vascular tissue found in the sporophyte. Fern sporophytes, like horsetails, have rhizomes. Leaves are now referred to as fronds, usually develop at the tip of the rhizome as tightly rolled up coils called fiddleheads, and they unroll and expand. The tight coil of the fiddlehead allows the delicate fronds to push out of the soil from their base on underground rhizomes. Once in the air, the fronds can unfurl without being damaged, and then fiddleheads are considered a delicacy in several cuisines, but some species contain secondary compounds linked to stomach cancer. Many fronds are highly dissected and feathery, making the ferns that produce them prized as ornamental garden plants. Ferns produce distinctive sporangia, usually found in clusters called sori. That is plural. Singular is sorus, typically on the underside of the fronds. Sori are often protected during their development by a transparent umbrella-like covering. Diploid spore mother cells in each sporangium undergoes meiosis, producing haploid spores. At maturity, the spores are catapulted from the sporangium by a snapping action, and those that land in suitable damp locations may germinate, producing photosynthetic gametophytes that are often heart-shaped, are only 1 centimeter thick, and have rhizoids that anchor them to the substrate. These rhizoids are not true roots because they lack vascular tissues, but they do aid in transporting the water and nutrients from the soil. The flask-shaped archegonia and globular antheridia are produced on either the same or different gametophyte. The multicellular archegonia provide some protection for the developing embryo. The sperm formed in the antheridia have flagella, with which they swim toward the archegonia when water is present, often in response to a chemical signal secreted by the archegonia. One sperm unites with a single egg toward the base of the archegonium, forming a zygote. The zygote then develops into a new sporophyte, completing the life cycle. The developing fern embryo has substantially more protection from the environment than a charophyte zygote, but it cannot enter a dormant phase to survive a harsh winter the way a seed plant embryo can. This is the life cycle again. And this has been your instructor, Scholar